Bioprecipitation. Bioprecipitation is one of the mechanisms employed by organisms for biosorption. Biosorption is the process where biological methods or biological entities like plants or microorganisms, including bacteria, fungi, algae, yeast, can be employed for removal of pollutants, including heavy metals and azotides, from the contaminated sites like water and soil. One important mechanism of this process of biosorption is bioprecipitation. Bioprecipitation involves the conversion of dissolved metal ions into insoluble forms through biologically driven chemical reactions. The microorganisms have different metabolic pathways. So they take up these pollutant molecules and they can convert these pollutant molecules into insoluble precipitates. Unlike adsorption or surface complexation, which are primarily physical or chemical methods, bioprecipitation results in the formation of solid precipitates. This precipitation is often catalyzed or facilitated by microbial metabolic pathways. This method is particularly important because it can be used for removal of heavy metals like arsenic, cadmium, lead, and uranium, which pose persistent environmental and health threats, including some are showing carcinogenic effects. They might cause cancer in human beings. Some are mutagenic. Also, these heavy metals can affect the reproductive system, immune system of the human beings. There are some conventional treatments like chemical precipitation and ion exchange, which can be employed for removal of these pollutants from the environment. But these methods are expensive and they often generate secondary pollutants, which again need to be treated. Biosorption is a cost effective and low waste solution. It is considered as sustainable solution and eco-friendly option. Also, it stands out for its stability and permanence. By transforming soluble toxic metals into stable, non-toxic, and insoluble forms, precipitation ensures long-term sequestration of the pollutant. In this process, the pollutants which are soluble in the ecosystem are converted to insoluble precipitates. These insoluble precipitates will remain in their insoluble form for a longer time. It plays a crucial role in bioremediation and wastewater treatment, especially in the scenarios where the goal is to prevent remobilization of the contaminants. These precipitates are insoluble, so they will remain in immobilized state and they will be removed from the environment. They cannot harm ecosystem anymore. The mechanisms of biosorption include adsorption, ion exchange, complexation, bioaccumulation, and bioprecipitation. In bioprecipitation, metal ions interact with biologically produced anions or functional groups that lead to chemical precipitation of insoluble metal compounds, such as hydroxides, sulfides, carbonates, or phosphates. Usually, these molecules or these functional groups are present on the cell surfaces of microorganisms. So these functional groups can also act as the binding affinity sites for pollutants in bioprecipitation method. The mechanism particularly effective in reducing metal bioavailability in the environment. While some biosorption processes are reversible, bioprecipitation typically results in permanent immobilization due to low solubility of resulting compounds. After interaction with the surface, the pollutants get absorbed in the cell and there they might undergo certain metabolic pathways to form these insoluble precipitates. Bioprecipitation is a process in which microorganisms induce or mediate the formation of insoluble metal compounds by modifying their local chemical environment. This often occurs through the production of metabolites like hydrogen sulfide, H2S, ammonia, or organic acids which react with metal ions and other pollutants to form precipitates such as metal sulfides, metal carbonates, or metal phosphates. For example, sulfur reducing bacteria that generate H2S, hydrogen disulfide. It can react with metals to form insoluble sulfides like lead sulfide, cadmium sulfide. This is not biologically controlled process but rather a biologically induced process where microbial activity indirectly leads to chemical precipitation. The microorganisms which are involved in this bioprecipitation process are having variety of genera including some sulfur reducing bacteria like D-sulfovibrio or ureolytic bacteria like Sporosarcina. A variety of microorganisms can contribute to bioprecipitation by altering the surrounding chemical conditions. Sulfate reducing bacteria, for example D-sulfovibrio species, are among the most studied as they generate sulfide ions. These sulfide ions are capable of precipitating heavy metals as heavy metals are having positive charges, and these heavy metals along with the sulfide ions form insoluble metal sulfides. Some ureolytic bacteria, for example Sporosarcina pasteurii, hydrolyze urea producing ammonia NH3 and increase the local pH, making overall environment alkaline, which leads to the precipitation of carbonate. So metal carbonates are formed and these metal carbonates will get precipitated. Some other microorganisms like phosphate solubilizing bacteria can release phosphate ions. These released phosphate ions cause metals like uranium or lead to form stable phosphate precipitates like uranium phosphate or lead phosphate. The biological reactions which are occurring inside these microbial cells result in the formation of solid phases that immobilize toxic elements. Types of precipitate formed. The nature of the precipitate formed during bioprecipitation depends on the microbial species, the environmental conditions, and the metals which are involved. Bioprecipitation method is also used for precipitation of certain other pollutants like dyes. The common precipitates which are formed include metal sulfides like lead sulfate, cadmium sulfate, zinc sulfate, which are typically formed in anaerobic conditions, where the organisms like sulfate reducing bacteria grow well in absence of oxygen. Metal carbonates like calcium carbonate or zinc carbonate can be formed. These carbonates are often formed by activity of ureolytic bacteria or photosynthetic bacteria. Some metal phosphates can also get formed like UO2PO4, uranium phosphate, FePO4, iron phosphate or ferric phosphate resulting from phosphate releasing bacteria. These compounds are usually stable, poorly soluble and environmentally benign. That means they are not going to harm the environment. These compounds being poorly soluble will remain in the precipitate form, that means unavailable to the ecosystem. It is not going to harm any of animals or plants or human for a longer time period. And as these compounds are stable, they are not easily converted to any soluble form. So bioprecipitation is a highly desirable biosorption mechanism. Factors that influence biosorption include different environmental factors, also the microbial consortia, and the nature of the pollutant which is involved. Several factors that influence the efficiency of bioprecipitation include pH, temperature, availability of counter ions like sulfide, carbonate, or phosphate, which are being available on the surface or by the microorganisms' activities, and the presence of competing metals. Microbial activity is also a crucial factor as only metabolically active cells can produce the necessary byproducts for precipitation. If the microorganisms are metabolically active in a very high rate, the process of bioprecipitation will be more efficient. Environmental conditions must support the microbial growth, like Some organisms can grow only in absence of oxygen, and they will show the activity of bioprecipitation in absence of oxygen only. For example, if we are using sulfate reducing bacteria, the environmental conditions should be anaerobic. If we are using some ureolytic bacteria that require oxygen, then the condition should be aerobic. Similarly, some organisms can show activity at neutral pH, some may show at low pH, that is acidic pH, or high pH. So all these conditions should support the activity and survival of the microorganism. Then only microbial cells can participate in bioprecipitation of the pollutants. And also in these supportive conditions, the microbial pathways, the metabolic pathways of microorganisms are active. Optimizing these variables is essential for maximizing metal removal through bioprecipitation. Environmental applications. Bioprecipitation has significant real world application. That means this is not only studied as a part of research, but this approach is being used for the bioremediation of the environment. It is also commonly used in the treatment of acid mine drainage, where it immobilizes metal ions like iron, zinc, and lead in the form of their sulfides. In industrial wastewater treatment, bioprecipitation is applied to remove toxic metals from effluents by forming insoluble precipitates. It is also employed in situ for soil remediation. That means in situ is the word or is the term which is used where the application is done where pollutant is present, where metal contaminants are immobilized directly within the soil matrix. Furthermore, bioprecipitation is used in nuclear waste management where it helps to immobilize radionuclides like uranium, strontium through phosphate precipitations. These applications highlight the value in both industrial as well as environmental settings. So, bioprecipitation is one of the major aspects. It can be used by applying the microbial biomass at the site of contamination, and it gives effective results.